Very good day to everybody. My name is Vasanth. I'm a NEET expert. I coach NEET aspirants who wants to achieve something very good. And I have an experience of 9+ years in teaching biology exclusively in NEET. Okay, preparing for NEET examination. And we aspirants actually train every NEET aspirants and medical, any other medical examinations, even engineering like JEE, SAT examinations. We train them in an extensive fashion, and that's what we are on. Okay, today in our class, we're gonna learn about a chapter which belongs to class 10. Okay, which is heredity and evolution. So let's get into the topic and learn something interesting today, shall we? Just allow me a few minutes. I hope everybody could see my screen. So this is the chapter. We are going to talk about heredity and evolution. So when you talk about heredity and evolution, these are two different important terminologies which you should be knowing when you talk about the genetic perspective and other things that is going to take care of a person's evolution with respect to time, over time. Okay, so that's an important aspect. So the topics that we'll be covering in this is introduction to heredity. I'll tell you explanations of what is heredity with few natural examples, and then real life examples, and then we will go on what is variation and its advantages, and an introduction on chromosomes, what are these, and then what are genes. So how are they different from each other, and how the Mendel, who considered to be the father of genetics, have actually contributed to this understanding process. Okay, and then what are the works that he has done, so which includes monohybrid and dihybrid cross, including the ratios. We'll be discussing them in detail. Okay, today's class is going to be fun. Okay, let's get into the topic, guys. So when you talk about introduction to heredity is where an inheritance, every wealth and the name that is accumulated by your grandfather will be distributed equally among your dad and your uncles and aunts, right? The same way that whatever your dad and mom have accumulated, it will be distributed equally between you and your siblings, right? So this is when it comes to wealth. So we have a quote which says health is wealth, right? So this is with respect to health. So a good health is always a good source of wealth for every human being. Now, the genetic composition that an organism acquires from the parent is actually one of the best combinations of characteristics that an organism can actually acquire, okay, which happens naturally, and that was made possible by the process of sexual reproduction. I hope every one of you have already gone through the chapter. Before coming to this chapter, the chapter name was sexual reproduction, and chapter name which includes the sexual reproduction part was how do organisms reproduce chapter. So in that we would have discussed about how it causes more probability or more chances of formation of new organisms and stuff like that. Okay, how they will be differing with respect to genetic probability, how they will be different from genetic material combination and stuff. So from whatever characteristics that was possessed by your dad will be transferred, not all of them, a part of it. So that will be from the father and this will be from the mother. So they'll be having basically 46, which will give rise to a haploid set of chromosomes by the process of meiosis division. Okay, and these gametes will be fusing, creating a new combination of 46. Okay, and this happens every time when an organism is born for the same parent. So that's one reason why the genetic material or the characteristic feature that actually that you can find between you and your brother or your sister could be different, or it will be different because of the probability and the differences. We are going to see all of them in detail now. Okay, now what is the function? Why this genetic composition, the probability, is going to create a huge difference? It's because the genetic material DNA, okay, which is going to be transferred from one organism to another in the process of reproduction. So they are responsible for the expression of characteristics because DNA will get to express, okay, they are called, the functional DNA is called gene, and then this gene will be forming RNA, and then RNA will be expressed into protein, and then the protein will cause the expression of the characteristics at a desired place and the desired location at desired time. Okay, so this is what is heredity. Heredity is where when a genetic material that we acquired from the older generations, okay, that they have acquired from the previous generation, our ancestors, so that gets expressed in our body, and that genetic material which gets expressed in our body is acquired from one generation after a generation, and it is passed along to us for a future of best quality that can be built in our body. Okay, so that's what heredity is. Okay, so you can see in this feature, organisms are passed from one generation to another is called characteristics. What is passed? Features of an organism, which means here feature means characteristics. So this process is happened because of the genes. So what are genes? Genes are functional part of DNA. So what do you mean by functional part of DNA? So DNA has both functional and non-functional part. So DNA has functional and non-functional part. We call them exons, okay, and introns. So this you will learn in higher classes in detail. Introns and exons. Exons are functional DNAs and introns are non-functional DNAs, non-functional, which will not be coding for any kind of characteristics. It might be having a hidden work, okay, hidden thing. Okay, so this process literally divides the genetic material, and that those exons will be combined to form genes, and these genes express to form RNA. RNA expresses to form protein, and these protein expresses to form the characteristics of an organism. So this is what heredity is actually. Okay, I hope everybody would have understood the heredity in detail. We have gone through this several times in this period of time itself. Now, after that comes variation. So during this process of heredity, during this product of inheriting the characteristics from one generation to another, it actually could cause it could actually be forming many variations. Variation means differences. Okay, differences. So what's a trait? Trait is what you see being expressed as a character in an organism. In my case, black eye color is actually one of the trait. Black eye color is one of the trait. So that trait will be expressed in here. Okay, so that could be influenced by not just by genetic material. It can also be through the environmental factors. So some of the characteristics could be influential. So let's say, for example, a same person who lives in the tropical region, his subcutaneous fat deposition will be minimal in the same case. So, but in the same case, when they are migrated to a much colder place, so their subcutaneous fat will be under the skin will be much higher in quantity. So that's a different kind of variation with respect to environmental factor. So and then there are so what is a variation? That is actually what is a trait. Okay, so traits are nothing but a distinguishable characteristics. Just as I told you over here, a recognizable feature is called a character or a trait. So that is a distinctive characteristics. Even a hair pattern, if it is going to be straight or curly, or else a black color, brown color, or even orange color. So the different types of hairstyle. So those are also characteristics. Anything which is recognizable, recognized to be a distinguishable or recognized, and it can be distinguished into different things, then it is said to be variation or characteristics, traits. So this variation is found, which means this characteristics will have several variations. Like the hair pattern, which I told you, curly hair, straight hair, and all those things are modified traits. So these modified traits would undergo a lot of changes in the future. So how is this caused? This is due to several differences that is caused to the genetic material themselves, in the process of expressing. So this is actually two types. These are actually categorized into two types. One is germinal and somatic. So germinal is where the differences will be caused in the chromosome wise, okay, in the chromosome or gene base. So if there is going to be a change in the gene, it's obvious that the DNA in which the changes are going to be, then RNA will also be having the change. When the RNA is going to have change, then protein formed will also be changed. When the protein form change, the characteristics expressed will be changed. Okay, will be changed. So this is going to have going to happen in the like a basic material itself, which we call this as gene. Okay, so next part is somatic. Somatic is like, you know, soma means body. So it is not the change that is caused caused in the genetic material that is causing the change in the characteristics. It is just the modification of some of the things in our life, daily life, that is going to cause an external change without having a change done in the genetic material. I repeat. So any changes which is happening to our body without having a change done on the genetic material is called somatic. So example, environmental factors like climate, food supply, interaction. So in the case of climate, like, you know, as I've already given you, people who goes to the, like, you know, much colder countries, they'll be gaining more fat, and then, like, you know, when they are going to be having more fatty food, so it is obvious then they'll be having so much fat deposited in the body. If they are gonna, like, you know, reduce the amount of fat, then it will be a reduced fat accumulation in the body, and then protein accumulation, and all those things will be, like, you know, totally somatic. Okay? It's all environmental factors. So these variations which could cause differences, there are actually categorized into 3 types based on how it is going to be effective on the society or an organism. So one could be useful. It could help an organism to survive and evolve, and then it can also retain its existence in this very world. So such variations are called useful variations. Okay, and the next one is neutral, where it will neither harm nor be useful. So it will not be taking any exclusive part in the process of existence of this organism in this very world. So that is neutral type. The next one, the third one, is the harmful one, where the variation, the change in the genetic material will literally kill or question the existence of the organism themselves. So we call them harmful. So there are 3 different types variation that could be the result which after the variation have gone through. Okay, genetic material have gone through the variation. So it could be any of these 3. So it could be useful, it could be neutral, or it could be harmful. It could be any of these 3. Okay, so this is about variation. So it could be advantages and disadvantages. So what happens? How can we discuss about the advantages of the variation literally? So due to accumulation of such variations, we can see accumulation of such variations, the body design could also be changed. Okay, a person who wants to become a lean person, okay, once after he reduces the characteristics and then he understands the environment and everything, so the body understands and accumulates the changes, and then it literally changes. So the body design will be changed. So that is a prediction that the fingers, okay, in this region will be, like, you know, will be like gone, will be like taken away. If the you know, variation is going to be continuing and in the process of evolution. So those are some theories which we can actually go through, and I'll give you more examples in real life, like, or some assumptions that you can make, which is actually understandable, and we cannot call it an assumption, then it is a realization. Okay, so there are future generations which leads to creating diversity. So this is actually another point which is very important. Okay, leads to creation of biodiversity. Why? Because every change in the genetic material will cause the diversification. So if this is organism 1, due to one particular change, 1 will be existing and 1A will also be existing. And after this, 1 again will be, like, you know, forming 1A, 1B. Okay, so there will be differences, and 1A will be forming 1A1, or it could be forming 1A. So likewise, if you see, there is a complete diversification of the characteristics when they are going to be kept on getting accumulated, and this will serve the like, you know, production of diversity in the organisms in a very fun manner. It will be, like, you know, very interesting manner. And example, so let's say we assume a scenario. Okay, there is actually a hot spring. Okay, a hot spring. In that hot spring, there is one particular strain of bacteria that is living, which is heat resistant. Okay, there is another type of bacteria which is actually salt resistant. So heat resistant bacteria and salt resistant bacteria are actually surviving in one place. Now, because of the sudden breakout of this like, you know, region, it causes the hot spring to increase temperature so much. So what actually happens is, in between this they will be producing a new variety called, okay, because of the interaction, the understanding, they will be producing a new variety which will be having both heat and salt resistant. Now, when the heat of the, like, you know, the fountain or the spring or the water resource actually increases, so the heat resistant bacteria will survive and the salt resistant bacteria will not survive, and the heat and the salt resistant bacteria will also be surviving. And when the heat goes higher and higher, there will be too much water evaporation that leads to sedimentation and accumulation of salt in the lake. So this bacteria over here, which is just heat resistant, will not be able to withstand the salinity resistance. So it is obvious this one will also die now. Okay, this one will also die now. So the only bacteria which is going to survive both the heat and the salinity resistance, who have accumulated both the characteristics, only can survive. So due to this variation that are being accumulated constantly in the body, it ensured the survival of the bacteria in this and then made it advantageous for a survival. Okay, this variation is an advantage. This is helpful. Okay, so, and I have told you several times, it is not always that the variations are helpful. Sometimes it could be harmful and sometimes it could be useless also. Okay. So there are different combination of characteristics that will alter the genetic material. So there are different types of things, methods through which the genetic material combinations could be altered. One is gametogenesis. Gametogenesis is where 46 chromosomes will be like, you know, will be like duplicated, and then they'll undergo a process called crossing over. Crossing over is something that you would have studied in class 9 itself, in the process of division. Okay, two homologous chromosomes will be undergoing overlapping. Two homologous chromosomes undergo overlapping. So this genetic material will be interchanged with each other. How? So due to this, it will be having something like that. So it is not necessary this crossing over should take place only in one arm. It can happen in all arms, or any one of it, or any two of it, or different regions. So this crossing over, you can see this genetic material has been switched and this genetic material has been switched over here. So this is not necessarily that has to be only this amount. It can be more than that, or it can be the whole arm, or it can be just a tip of it. So in this way, that gets to be random. So crossing over happens to be random during the process of meiosis. So in this case, the gametes formed will also be having differences. All the gametes formed will be having differences. So the gamete differentiation, okay, the chromosome separation will be causing the first step in the cross of variation, and the crossing over makes it more complex in the process of accumulating variation, differences in the genetic material. And now what happens during the fertilization, we will not know, because male usually produces 400 up to 400 million sperm. Okay, in that 400 million sperm, any one of that sperm which with unknown combination will be fusing with the egg that is available in the female reproductive system. So this gives a probability of 1 in 400 million. Okay, so that kind of variation is also caused. So these are the three main facts where we can see variations could be, like, you know, made so much possible in genetic material perspective. And other cases are accidental, where the genetic material could be added on top of the already existing one. So this is also possible, or deletion, okay, or the genetic material could go inverse. So these are some of the chromosomal changes that they will undergo, which could also cause abnormalities and like problematic creation. So this could be problematic sometimes, or it could create also like, you know, a betterment, like, you know, in the process of variation. Okay, so we'll go to the next part. So next part is we're going to study about chromosome and gene. So what's a chromosome? Chromosome is a complex structure. So what is this complex structure contain? Complex structure contain proteins and DNA, deoxyribonucleic acid. So these two actually are responsible for the formation of chromosomes. So why chromosomes are formed? Is it available in the plant or in the animal or in the microorganisms? In which one? It is available in almost all organisms which are eukaryotic in nature. So why is this important? Why is this important? This chromosome is a compressed structure of DNA material. So I'll ask you a question. When you are going to shift from one place to another, you have a lot of glass items. How do you tend to transfer them? You pack them nicely with bubble wraps and then settle them in a a box, cotton box, and then you tend to transfer them, right? So what if you don't do that and then you transfer them? You tend to break it a lot. Okay, there is a lot of probability that it will be broken. Now this chromosome is made up of genes or DNA. So this DNA is very fragile in nature. DNA is very fragile in nature. Thus this fragility could be overcome when it is going to be wound around a structure that going to make the genetic material compactly wound to form the chromosomes. So that bounding around part, okay, happens on the proteins. So I have a clear picture with which you can actually understand the process very clearly. Okay, I think you can all see this. So this is the nucleus. The nucleus, upon division, only during the process of cell division, this chromosomes will be formed. So the chromosomes are made up of these kind of segments, and these segments are called nucleosomes. So nucleosomes are made up of histones and DNA. Histones combine together to form the bobbin, okay, or the spool. On top of it, the DNA will be wound around. So in the DNA, any segment of DNA which is going to be coding for a particular character, then we call it as genes. And these DNA are made up of nucleotides. So nucleotides are guanine, cytosine, adenine, thymine. So these are also called purines and pyrimidines. They are grouped into two, purines and pyrimidines. So adenine and guanine are called purines, and thymine and cytosine are the pyrimidines. I repeat, adenine and guanine are the purines, and thymine and cytosine are the pyrimidines. So it is a complexity that A always bonds with the T, and the G always bonds with the C. So you can see that A and C, A and T and G and C, bond with two different. I have put different lines, which means A bonds with T with 2 hydrogen bonds, and G bonds with C with 3 hydrogen bonds. So because of their chemical nature, okay, so they are already weak. So it is very fragile in nature. To make it safe. Okay, so the genes will be the genetic material will be wound around the protein structure called histones, and that forms a nucleosome after when the DNA is completely wound around the histone proteins. Okay, so this is actually the structure about chromosome and genes. So these inheritable characteristics through chromosome is actually transferred through gametes, and these gametes contains chromosomes, the genetic material, and that is made up of DNA, and DNA contains gene, and the gene is responsible for production of proteins when they are expressed. So from DNA, a part of DNA with this gene which is actually been done, and in between there is a process where RNA is also formed, and from that RNA protein is formed. So from DNA to RNA, this process is called transcription, and from RNA to protein, this process called translation. So this is actually how the expression of characteristics takes place. So the protein formed will undergo secondary modification, which we'll be studying in higher classes in 11th standard under the chapter biomolecules. So this modification will actually make it functionable. Okay, so that modification will make enzymes, okay, and or even functional structures. Okay, so it could be many things, or signaling machineries. So it could be any of it. So this is the purpose of the genetic material. The genetic material is like a book which actually has everything present, every important stuff that is written over, so that it can be taken out and then read, and then knowledge is acquired, and then the work can be done, and a structure can also be prepared for doing the work. Okay, so that's the process of, that's about chromosome and genes. So when you talk about genetic material, so we cannot leave Mendel. Okay, so Gar Johan Mendel, that's his full name. Gar Johan Mendel, that's his full name. He was actually a monk. So this monk was actually actively working on the cathedral. So this, when he was going to the cathedral, so he saw that there were tea plants planted in that region. So in that region, one tea plant was growing very tall, okay, and another one was dwarf. We don't call short, we call them dwarf. DWARF, dwarf. So you need to be very careful when you say it. And he called the people and then asked like, when was this plant? Because he saw himself that he said he got an answer that it was all planted at the same time. So there is no difference in when they are going to be, when they are planted. There was no difference between the seeds. They were like same time in the same place and everything. But how can these two same seeds or like same type of seeds that is going to express two different characteristics? One plant tend to be tall, another one tend to be dwarf. So this literally sparked him. Okay, sparked in his head. So that is when he started to work on like what was actually causing this difference. So he assumed that the thing which was actually causing the difference is factor. He called it factor. There is some factor that is influencing the expression of a characteristics of an organism. So he called that factor. So later this factor was identified to be gene, and then the factor is nothing but the gene which has a modified genetic material that is going to express a different trait of a character. So that is what Mendel's contribution was. So it was Mendel who actually tracked in lots of things. See, he was able to track which one was dominant, which one was recessive. In the dominant one, like, you know, how it is dominant and how it can exist, and how like, you know, after it has been like combined together, how they are going to be segregated, and all these things could be learned about. So that's what we are going to learn in the upcoming things. So for knowing all these things, someone have to experiment. So Mendel did the experimental part also. He did a complete experimental approach. In this experimental approach, like, you know, he noted down, or he actually was like, you know, pretty concerned about few things. One is he selected only pure plants which are like, you know, purely tall, and then another case which could be purely dwarf. So this, by doing this, he chose the desirable character which he cannot be fooled with at all. So that is one like, you know, thing that he always looked into. The next one is the first generation formed will be like, you know, bred by fusing the pure breeds. Okay, the cross between the pure breeds of example, the tall plant, the pure tall plant, and pure dwarf plant. So this is process is called hybridization. This process is called hybridization, crossing two different trait plants. Okay, so how did he actually manage to do that? He actually managed that. So there are two different flowers, right? Flower 2 and flower 1. So in flower 1, he have emasculated. I repeat, emasculated. What is emasculation? Removal of anther. Removal of anther. And then in the capital T and capital D, he removed the female reproductive part. Instead of removing the female reproductive part, he again emasculated and collected the anther. So by this process, the anther that is collected from the second plant will be used on this plant, this first plant, and the anther which is removed and collected, will be used on plant 2 to actually undergo pollination. So by doing this, when he does the pollination for the first one, he usually covers the second one with bag so that no other pollinating agent could actually infest this plant with any other pollen. And the same thing after pollination, he will also bag them. So this process called bagging. So removal of anther, bagging, fertilization with collected pollen, and then bagging. So by doing so, it restricts the influential influential work that is done by the natural pollinators. So now when he has done the bagging part, everything is under his control. Nothing goes out of his knowledge. So that is what is happened over here. And the next one is when the first generation is formed by the hybridization of pure plant. So the second generation that is formed from the first generation would be selfing of the F1 and F2. So when you say the first generation is capital T and small t, the gametes formed will be capital T and small t's. So when they are crossed, we get a capital T small t plant. So the second generation will be formed from the hybridization of these plants. So when we will get two types of gametes each plant. So then when you get it, it will be something like this. So it will be capital T, capital T, capital T, small t, capital T, small t, small t, small t. So here you can see this is tall, this pure plant, and this is dwarf pure plant. And this is a hybrid plant. These two are hybrid plants. So there's a probability of 3 tall plants, okay, and 1 dwarf plant. So this is what is explained over here. So he raised the second and subsequent generation by selfing or hybrids. Selfing or forming hybrids. So F2 generation, F3 generation are self-fertilized with their own generation individuals. So that is what I have done an example for you. So this is actually was the approach that is taken care by Mendel himself. So in this case, he actually tried to explain monohybrid cross. So monohybrid cross, mono means single character. Hybridization done with the help of single characteristics. Okay, single characteristics. So hybridization done by single characteristics. So this is actually a trick which I always tell you. Most of the cases, if you separate the term into 2 or 3 or parts, many parts, as many parts as possible, that is making sense, and then read it in reverse, you will understand the real meaning of the word. Okay, what does it do and stuff. So here he did the process of genetic mixing up. Okay, 2 individuals. So the 2 individuals were capital T, which was like, you know, the phenotype. The phenotype is like anything which is actually seen as a character that is expressed. That is called phenotype. And the genotype is the genetic constitution which is causing the expression of the character. The genetic constitution of it is the genotype. So if you see, tallness can be capital T, and the tallness can be capital T, small t. So if you see the growth of this plant, both of them will be tall. Okay, the phenotypically they both will be same, but the genetic constitution wise they are different. How? Anything which has same is called homo, and that is actually enclosed in one thing. So homozygous. Sorry, just a second. Homozygous. Zygous. And when they are going to be 2 different, so it is called hetero, H-E-T-E-R-O, hetero, zygous. So this is one thing. And when you talk about the Mendelian cross, okay, so that's what we call it, Mendelian monohybrid cross. So crossing a plant with 2 different expressing traits for the same character, I repeat. So the character here is height. Height is the character, and the character can have more than one trait. So it could be an expressing trait, which could be tall and dwarf. Okay, so now this is different. This is different. Now we'll talk about a different thing now. Okay, what's an allele? We're going to learn about this term again and again over here. So allele is the set, the chromosome or the genetic material that is going to be responsible for the expression of the characteristics. So this actually contains two different alleles. So in this heterozygous plant, the alleles that are present will be two different types, and this is going to be same allele. So if the alleles are going to be same, that there is a homozygous. If the alleles are going to be different, then it is called heterozygous. So this is the simple way to understand the process. Now this is the way that you have to apply for the process of monohybrid cross representation. So you have to write everything in detail. So this is parent P1 cross, and which include a tall plant and the dwarf plant. That is what is the pure breed. So their phenotype is tall and dwarf. What about their genotype? They are pure breed. So it is supposed to be capital T for tall and small t for dwarf. So when they are when they are allowed to undergo reproduction, the individuals will fall gametes because it is of same allele. So only one allelic form can be taken as gamete, and the same thing is done for the pure breed on the other side as well. So upon fertilization, it forms an individual which is heterozygous in nature, which is capital T and small t. Okay, so that's the F1 generation. When the F1 generation, all of them were tall. So then he realized, like, why is it one tall, another one is dwarf? So it should have something to do with it. Okay, so because we had a cross between a tall plant and a dwarf plant, but the expressing characteristics were only tall. Okay, and then later he was able to understand, like, few things which we'll be discussing in the upcoming videos. Okay, so in this he have actually performed the for F1 cross. In that F1 cross place, the individual of the F1 generation will be allowed to cross. So that will be capital T small t, capital T small t. So they'll be having two probability of gamete. So capital T will be a gamete and capital small t will be a gamete in both the parents. So upon crossing, they will get a homozygous tall, a heterozygous tall, another heterozygous tall, and then a homozygous dwarf. So here, the thing that you need to know here is the phenotype of the first three plants are tall. Okay, because they have the capital T over here. So this is where he realized that there was one particular allele which was expressing or dominating over the expression of another characteristics. There was dominating. So this is what he realized. Then later he understood that it was the ratio. It was expressed in the ratio of 3:1. Okay, 3 tall plant and 1 dwarf plant. So that is the phenotypic ratio. Phenotypic ratio. So what is genotypic ratio? Genotypic ratio is where the genetic constitution of the organism, so which is 1:2:1. So one tall plant that is homozygous, two tall plant that are heterozygous, and another dwarf plant, one dwarf plant that is homozygous in nature. And that's what is the conclusion that he has come up with the monohybrid cross and ratio. Okay, he also understood many things that we'll be doing in the upcoming videos. Okay, so after, I hope every one of you understood the monohybrid cross and monohybrid cross and ratio. So the monohybrid cross and ratio, you have to box the parents and you have to circle the gametes. So it is mandatory. Okay, this is how you have to learn. This is how it has to be represented also. Okay, I hope you should not get confused. Next is Mendel's contribution with respect to dihybrid cross. So dihybrid cross is right. So I've already told you, hybridizing two different character, not trait, two different character. Okay, here we are going to take two characters. One is seed color and seed shape. So seed color includes yellow and green, and yellow being dominant, so you are taking that letter, okay, and green being recessive, we take the smaller letter. And seed shape, there are again two. One is round and wrinkled. So round being dominant, we take it capital R, and wrinkle being a recessive, we take small r. Okay, so this is the characteristic, two characteristics. One is seed color, another one is seed shape. So dihybridization, hybridization of two characteristics, okay, with four different expressive traits. So now when we are going to take the same thing, so there will be pure breed, that will be crossed. So here you can see the round yellow. So how do you write it? It is just that capital R, that's the pure round yellow seed, and then the next one is pure wrinkled green seed. Now when we are going to cross them, okay, now when we are going to cross them, so they are going to form capital RY, that's the gamete, form. So here also their gametes are going to be formed, will be small RY, which is green wrinkle. So upon fusion, it forms capital R, small r, capital Y, small y. Because of the presence of two dominating allele, this seed will be round and yellow. So both the characteristics will be expressed. Now, the second part, the F2 generation formation, will be selfing of the first generation. Okay, selfing of the first generation. So here the gametes formed would be like, thoda sa complex, like it will be complex a little bit. So now how do you determine that? So that is what this segment actually represents. So it will be capital R with capital Y, okay, capital R with small y, small r with capital Y, and small r with small y. So by doing so, okay, we get a combination over here. So you see capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So we do that on both sides. So this square shaped structure, we call it as Punnett square. Punnett square. So this Punnett square is actually an easy way to acquire the ratio very easily. So by crossing, so the column and the row, so the gametes that is found in the column and the row could be written united. So capital R, capital R, capital Y, capital Y. So capital R, capital R, capital Y, small y. Capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. So likewise, if you keep on writing, you'd be getting this to be round yellow, because capital R, capital Y present, capital R, capital Y present, round yellow, 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 capital R, small y, so there is no capital Y, so it will be green in color, round green, and this will be capital R, capital Y, which will be round yellow, capital I with no capital Y, so which is round green, and capital R, capital Y, which is round yellow, capital R, capital Y, which is round yellow, and small r, capital Y, which will be round, which will be wrinkled instead of round, so it will be wrinkled yellow, and then here it will be small r, capital Y, so it will be yellowish in color, but it will be wrinkled in shape. And capital R, capital Y, round yellow, capital R, small y, so round green, small r, capital Y, small r will be like wrinkled, and then the capital Y represents yellow color, and then small r, small y, which will be wrinkled and green. So in this case, if you calculate all the round yellow, 1, 2, 3, 4, 5, 6, 7, 8, 9. So the expression of the characteristics, we say the phenotypic ratio would be 9 round yellow, okay, and then round green 1, 2, 3, that will be 3, and wrinkled yellow, so 1, 2, 3, so that will be 3, and the last one will be the wrinkled green, that will be 1, only 1. So 9:3:3:1. So 9 plus 1 is 10, and 10 plus 6 is 16. So 4 rows and 4 columns, 4 into 4 is equal to 16. So that is also the phenotypic ratio. Now, the genotypic ratio for this is really tough, but it is easily acquired. It is easily acquired. So that requires for you to check how many of them were capital R, capital R, capital A, capital A. So there are only 1. Okay. And then capital R, capital Y, and small y. So likewise, if you keep on adding, it will be giving you the genotypic ratio, and you can use that for things. So I didn't want to give it away the answer. I just want you to work out how it is different. Okay, I'll give you a table at the at requirement when it is required. You can ask me in the class. And now this is the Mendel's dihybrid contribution. So we are going to study what we are going to learn, what we have learned from monohybrid and dihybrid cross. So he has framed actually 3 laws, which is called laws of inheritance, which include law of dominance, law of independent assortment, law of segregation. Okay, so these are the 3 different laws that he has conceived from working out the monohybrid and dihybrid cross, which we'll be seeing in the upcoming video. Okay, so now we'll have few questions to discuss. And what is monohybrid? As I've already told you, mono means single hybrid. Single characteristics are used for hybridization, and the ratio represents how cross characteristics are expressed. So in the case of monohybrid, there are 2 types of ratio. One is phenotypic and then genotypic. So phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. Okay, so that's the answer for Mendelian monohybrid. We have studied that in detail actually. And what is heredity example? Okay, so heredity is a place where the genetic material of the parent is actually passed on to the next generation, and this passing of genetic material will actually have a lot of possibility for the organisms to survive. Will give a lot of possibilities for the organisms to survive. So, for example, a person who is always having the possibility of having a genetical disease. So it could be transferred to the next generations. Okay, normally. So this is heredity. So heredity is where it could be bad, it could be good, it could be useless, as I've already told you. So heredity in inheritance, where good characteristics can be inherited, bad characteristics can be inherited, and useless characteristics can also be inherited. So this is heredity, and I've given you an example to that. Thank you so much for joining with me for such a sweet class. And I hope everybody have enjoyed it. So if you have any questions, we always welcome questions, and we will be delighted to answer. We have exclusive team of faculties who are very experienced in actually giving away answers that fulfills the TIE's requirement. And please don't stop from just enjoying our videos. Join us live in live classes and learn and enjoy the classes. Okay. Thank you so much, guys. Have a nice day.